I'm really happy to be talking about this project called SwabSeq, which is really a very crazy project in everyone's life who was involved in it. I talked a little bit about it last year, and so there's because some of it's so good, some of that, it's still there, but a lot of it's new. So if you were here last year, please bear with it. Okay, so what is SwabSeq? So this actually came from what now feels like a completely different time, but if people remember in March of 2020, what was going on. So this is kind of some news from March of 2020. This is kind of the headlines. This is just some of them. This was another one, right? I don't know if people remember like when Tom Hanks got COVID in Australia. That seemed to be a really big moment at the time. And this is, at the time, this was a scandal, right? That 58 tests went to MBA team rather than people that were sick, and this was like a big, national news at the time. California was made actually $1.4 billion investment to try to build a new lab because there's nothing available. So, so in that context, a group of us basically got together, and we were all sent home because we couldn't. They closed the university, but they made an exception. If you wanted to work on a COVID project, you were allowed to go back into the lab. And so a group of us at UCLA decided we were thinking like, well, we have all this equipment that we have at UCLA. We can do all the testing that we need. And not only that, we have these sequencers that maybe we can try to develop a test based on genomic sequencing. So that's really how this project was born, and we developed very, very quickly, pretty much by the end of April of 2020, we had the technology working. It's called SwabSeq. It's a new technology. The idea is that it is a sequencing-based COVID test. So the idea is that in the first step, in the PCR that amplifies a piece of the COVID genome, it adds a molecular barcode. Then all the samples are mixed together, and then they're run on a sequencer, and then a software then deconvolves it and makes the calls. And so there's kind of key differences is that it's based on genomic sequencing. It doesn't require extraction because the sequencing is inherently more accurate. It uses this barcoding, and the readout is sequencing, which is digital, versus fluorescence, which is analog. And actually, last time I gave the talk, somebody pointed out that sequencing is also fluorescence, but this is fluorescence converted to digital rather than fluorescence converted to analog. So that's the difference. Okay, so just a quick, just a kind of a toy example. What do I mean by barcodes? So the barcodes. So here's the idea of a barcode. So we have the amplicon, which is something that's the PCR is amplifying, and the way that it's set up is that the process adds a barcode to every sample. So, for example, individual 1 could have these 5 A's added to it. Individual 2 has 4 A's and a C, et cetera. And it's not exactly how this works, but the idea is that this is added in the first step of processing, and then after this, all the samples are mixed together, but they've already had their barcodes attached. And then when we sequence them together, we can tell which sample came from which individual because of the attached barcode. And so that's the really key benefit of this technology is that we could scale this almost arbitrarily. And so we were, at our peak, we were sequencing 6,000 samples. We were actually analyzing 6,000 samples at a time on one instrument, right? This is a huge difference compared to traditional technologies, which usually do 384 in a PCR machine. And so you could imagine that you need a lot of PCR machines, right, in order to be able to do that many samples. So just a few more details. So this is what it looks like. So this is the amplicon, the primers. This is a target on the sequence, and we have these two barcodes that we add, and they're actually the primers themselves have the barcodes in them. So every individual uses a slightly different, every sample has a slightly different PCR primer, and that differs based on the barcode.
And that's done in the first step of processing, and it allows for all the sequencing together. So that's like one key idea. The other key idea, which also is kind of something that really only makes sense with sequencing, is that we add in what's called a spike in, which is a synthesized sequence, which looks just like our target, except for it has 6 of the bases reversed, and we add in a known quantity into every sample. Because we're sequencing, we can very easily distinguish between what's a real and what's the synthetic, because we can see what those 6 bases are. And so what that does, and so I'll give you an example why it's important. So here's actually a plot that shows kind of the limit. It's called a limit of detection plot. So the x-axis here is different concentrations of kind of synthetic virus that's added to the sample, and every dot is a sample, and the y-axis is the number of reads that you see of the target sequence. And so you can see that it's kind of working in the sense that you see a trend, right? So as you get more and more virus, you see more and more reads. This is on the log scale. But there's also a problem because if you're going to classify which ones are positives and negatives, that would be a horizontal line in this graph. And they're not, clearly you'd have a bunch of false positives or false negatives depending on where you draw the line. There's no real good place to draw the line. So overall, this is not going to be that accurate of a test. But if instead of looking at the total number of reads, you look at the ratio of the target versus the spike, the data is much, much cleaner. And the reason is because typically, like for example, down here, you see that there's a couple samples which are very positive, but they look negative. It's because there's actually something in the sample that's preventing a PCR reaction from happening, and so that you don't have a lot of amplification. But that also prevents the synthetic sequence from being amplified. So then those two samples, these are the same samples, those two samples would be, are much higher, right? So that's the basic idea, and it results in a really accurate test. We have to, like, part of doing a clinical diagnostic is you have to come up with kind of a very firm way of deciding what's positive, what's negative. And so that's what, we came up with this actually in advance of seeing any data, right? You have to kind of come up with a plan. And so this is kind of this decision tree, looked at counts of reads and ratios and decided what's positive and what's negative. So that's what we started with. And so that's what we started with. And so we actually approached the UCLA leadership, and this is in early May, and of course they're the hospital's closed and there's COVID patients in the ICU, and so they have a lot on their minds. But we came up to them and we said, hey, we've actually solved the COVID testing problem, and we showed them this data, and we said, let's kind of go with this. And so they now thinking about it, I realize that they were probably quite skeptical in this meeting, but they actually decided to, they told us, they said, like, look, we believe you that the genomic technologies work, but the logistical, turning this into an actual test and collecting the samples and processing new technology, even with the current technology, is really challenging. But if you want to try it, and you don't, we're really busy, so we don't have a lot of time to help you, but if you want to try it on your own, you're welcome to try. So that's kind of how this project started, and they actually, the other thing that happened was that they're planning on building this immunology institute at UCLA, which now they are going to build, but they were talking to donors for a long time, so they had 3 floors of wet lab that were completely empty, waiting for the $1 billion check to arrive. And so when the pandemic came, they were like, oh, we actually do not want you to do COVID testing in the human genetics building. That seems like a really bad idea. But we have this empty labs that you can use. So they actually gave us these labs. So that's one of the things we need: dedicated space, money, all these things. And so they gave us this beautiful like floor of a building with nice views, and so that's kind of where we started building our facility. And so then the next thing that happened is that, if you remember, there was a huge rush on supplies, so you couldn't really get any supplies. So then what we did was, so we were talking to these vendors, and of course the vendors, the salespeople are always talking to you, even if they have no stock. They're pricing, having you issue POs, you send them POs, and then only when you pay for it do they say, oh, we don't actually have the stock. And so we asked for, we thought 20,000, that seemed like a large number of supplies, 20,000 tubes, that's what we wanted to do for our thing. And they said, and they do go through the process, they say, oh, we actually can't, there's so much demand, we can't send you 20,000. We can either send you 800,000 or zero. So the minimum amount is 800,000, right? So then we went to our leadership and we said, okay, so here's the situation. Either we can get zero or we can get 800,000. They're like, how much is that going to cost? $1.25 million. They said, let's think about it. They let us think about it. And so finally they said yes. And so we ordered, all these supplies. And so this is kind of what the timeline of what happened. So April 2020, we came up with the technology and showed that it worked. We moved into the lab. We submitted our regulatory, like FDA application in July. At that point, you needed a FDA emergency use authorization to do the test. And we were actually able to start operational in the middle of July of 2020. And we were mirroring the testing that the campus was doing, but we didn't have the license until the end of October, so we couldn't really do anything. But in the meantime, we hired staff and our supplies finally came in, and we finally got the license in October. But I just want to give a little bit just perspective. So, in July, we were both submitted our application. So by that time, we had all the data and everything that we would need for the license. And in July 13th, we actually we were actually doing operational testing, but mirroring, because we couldn't return results. But it took all the way through November 9th for us to actually do our first deployment. And this is almost completely because of regulatory waiting. And a weird thing happened during this time. And so the thing is that we submitted our application on July 2nd, and of course it could have been approved the following week. It was actually approved in October 7th, but it could have also been approved October 7th, 2023, right? There's completely no insight on when it could be. And so the UCLA was like, well, we have students coming back, and what are we going to do? So UCLA actually signed a contract with another company to do all of their testing, and that wasn't, and it was kind of binding, so they couldn't get out of it. And so because they thought at some point that our license was never going to come, of course our license came a week later, and so at that point we had a very interesting situation, because we just spent all these millions of dollars, per building this lab, and we didn't have anyone to test for because the campus signed a contract. So at that point the campus, and so we're having it was kind of frustrating because we're having these, leadership meetings every week with them, and they of course didn't tell us that they were thinking about this, but they decided to do this. I don't blame them because they needed the testing. So then they helped us become a vendor, because we kind of convinced them that, well, we have all these tests and there's a lot of demand. Why don't we test for other universities and we'll try to make back our debt, right? And so they were very skeptical, but they let us do this. And so anyways, we kind of came up with plans for doing, we also had to kind of, at this point, start not spending a lot of money. And so we came up with many processes that are very efficient and require very little staff to do that. And also just kind of bootstrap. So one of the things that was at the time very hard to get and very expensive were these saliva collection devices. They were like $5 each, and they're hard to get, and we couldn't really find them. And so actually Valerie Arboleda is one of the key people in the technology. She actually realized that perfume funnels fit perfectly into the tubes. and we can buy them on Amazon. So we've got started. Our first round of our saliva collection devices were, we bought 20,000 perfume funnels on Amazon, and that's how we got going. And once we realized that those worked, we actually had them kind of manufactured. And so anyways, we got our supplies, and this is kind of where we're going. So we actually ended up testing for UC Santa Barbara, UC Irvine, Caltech. We actually, the health system, UCLA Health, we started testing from. Cal State's, just many other universities. We ended up testing for the school district, and at this time, and then we ended up taking over the UCLA testing in June, kind of when COVID seemed to be over the first time, and there was no positives, and so and UCLA actually wanted us to take over because. First of all, we didn't have any minimum contract like the other vendor, and they thought it was over. And also that they wanted us to do self-collected testing machines. Also at the same time, we ended up getting this NIH RADx, which is this organization that funds new COVID technologies. They kind of heard about us, and they decided to fund us to expand our facilities because we're one of the only new technologies. And so, but just to give you a sense, like through this process, there's like limitless numbers of problems. Like for example, we wanted to do COVID testing in the vending machines, and if you look around, there's actually several of them still on campus, and now they're actually open to anybody. So if you want to use this, you're welcome to use it. So this is the idea, but this was the reality, right? So all of the tests would get stuck and they wouldn't come out, and then people would try to like tilt them backwards to shake them, and the whole machines would almost, like, it was very dangerous. So actually, we didn't solve this problem. We were thinking of trying to make 3D printed funnels inside the machine to help the test come back, but that never got up. Actually, the campus actually came up with a great idea, which was that they put weights in the test. But weights are expensive, right? So instead, they put candy in all the tests, right? And so there's candy in all the ones that were in the vending machines, but there wasn't candy in all the ones that weren't in the vending machines. And obviously, they were buying the cheapest candy, so there's like different candy everywhere. So on the UCLA Reddit, there were all these discussions about which vending machine has better candy. Why is there candy on these and not these? But from our perspective, we were like, we don't say anything, from our team, right? So. Yeah. Well, because you're not supposed to have the candy before you give the test, right? So like we decided not to say anything, right? So as a policy. And so then we got these, the funds to expand our facility. At the time we got it, we really thought that this would not be needed. And that allowed us to buy automated systems to do the testing. And then, of course, what happened was that, we ordered this equipment, and then the Delta variant comes in, right? And because of that, this university wants to reopen because we were pretty much closed for the first year. And they decided they want to test everybody twice in the first week, and then all the other universities that we're working with also want that. So we have this huge demand in early 20, in the beginning of the year, 2021, like September 2021. And so, but we're thinking, oh great, well we just got this money to buy all these robots. This'll save us, right? But of course, what happens is that we do the POs, we order the robots, and then the robots are only going to arrive in November. So we had to come up with a different system to get through there. And so actually somehow the lab actually figured out a way to do about 6. So we were able to do 1536 samples on a run. So they were able to do about 6 of them. So it's like, over 9,000, close to 10,000 tests a day. And so they were able to do this, but at a really crazy effort. And so, and this is kind of what it looked like. There was the demand was even higher. So it was actually a very difficult, just very difficult time to try to get these, to get this through. So we actually came up with this idea to like increase our throughput, which was that. So in general, we had enough we only really had 1536 barcodes. That's all we had. Those are all the ones that we had because we didn't think we would need more, right? And it actually getting barcodes and making sure that they're produced correctly and validated, don't interfere with each other, is like a huge project. It's like not something, it's like a month-long project. And obviously if you added more barcodes, then you'd have even more potential interactions. And so that's something that's just not realistic in any you know, in any like short period of time. And so, but we actually had such problems with getting our barcodes that we wouldn't order them plated. We would order like raw stock of the primers, and then we would mix them ourselves. So we would order basically, these are all the plates for, you know, we would order like the reverse and the forward primers, and then we would mix them. And we do that both for the target and for the, you know, we have a control gene. So we'd order 16 plates at a time, validate each one separately, make sure that they're okay, communicate with the vendor if not. So we essentially had them all in pieces. And so what we realized is that because they were this way, we could essentially mix them together in kind of different ways to actually get, instead of just 1536 barcodes, by mixing them, the plates, you know, by forward and reverse in different ways, we can get up to you know, over 6,000 samples. And so that's what we did. And that's a combinatorial indexing. Technically it's not as good as kind of the unique, you know, you essentially have half of your barcode will overlap with, you know, 4 other samples, right? But it worked, it worked well, and we were able to get, you know, 6,000 samples at a time. And then using that, we were able to have, so these green runs are runs where we had, you know, more than 1536 at a time. And eventually, once we got the whole thing working, we were able to get, you know, 6,000 runs a day. So we would actually be getting like close to 12,000 samples in the lab, and our lab would only run 2 sequencers, right? One to do 6,000 in the morning, and then one to do 6,000 in the afternoon. Okay, so this is what, with all the automation, what things look like. You actually, better if you want to see the lab, we can show it. We can show it to you this week if you want. So then Omicron came, which is good because, you know, our automation only came after that big push. But then with Omicron, we ended up testing, UCLA ended up mandating testing pretty much continually for the whole rest of the year. So we ended up doing close to 2 million tests. And so there's other interesting facts. So here's actually, this is the kind of thing that we learned. So for example, this actually, it's only these first 7 columns. These are samples, and kind of the red color is signal. So one of the things we had to do is we had to try to do these interference tests to see like what would interfere with the COVID test. Okay, so these are all the different options. Okay, and so one of these columns has completely no signal. So there's something that completely interferes with the signal. So can anyone guess what it is? So if you wanted to have an inconclusive COVID test, right? You would take one of, like, you know, so so essentially there's one item here that completely inhibits PCR, completely. Huh? No. Yeah, it's a toothpaste. Yeah, toothpaste. Yeah, and in fact, just because, you know, we were like, sometimes you're curious, right? So we wanted to answer a question which we've always wondered, which is that, you know, the organic toothpaste they sell at Whole Foods, does it really work, right? And so we got some of that, and we tried the same experiment. And actually, it doesn't. It still inhibits, but it's less. So it doesn't work as well. Yeah, clearly, based on this experiment. So, okay. So then what's anyways, what's next? So we deployed an accurate, scalable $10 PCR test. And so that was our original goal for the project. And this is actually, I think, the first time in my entire career that I've ever started a project and actually feel like I finished it, right? Like I don't think there's any project that I've ever finished, right? But this one, I think I feel like we solved that problem. That's finished. So the question is really, what's next? And so the next thing is that, you know, we are using sequencing in SwabSeq, but it's, you know, I mean, it's not really utilizing what's interesting about sequencing. We essentially using the sequencer to do the PCR test more efficiently. That's all we did, right? But obviously the The real power of sequencing is to look for, any virus or any pathogen, et cetera. And so, so we actually got funded by BARDA to do this. And so I wanted to kind of say, like, this is kind of a high level. What is the different approaches? So if you have, essentially you have a pool of virus, like if you get a sample, like a nasal sample, you have huge amount of RNA in there. Almost all of it's human, and you have a very small amount of virus. And so typically, people will use PCR to amplify the virus, and then then it makes the majority of that sequence, the virus RNA, you can see it. But you have to know what you're looking for. So that's one thing. So the other approach is you can try to use depletion to eliminate human RNA. And so this is very commonly done for like ribosomal RNA, but then also now there's technologies that can try to target using CRISPR, like all of humans. So that's also an approach. Or there's even another approach, which is actually works even better, which is just the sequencers have gotten a lot better, and so just sequence a lot, and then eventually if you sequence enough, you'll see it. So, and there's huge amounts of issues that affect how you do this because, for example, nasal samples are not like blood samples or cultures or anything like that. They're very low quality, and they affect, everything. Then artifacts, your processes really will create lots of artifacts. So this is just to show an example, very low quality. So typically, when you look at RNA from a nasal sample, swab sample, you can't detect it. Like if you try to do what you normally do to do sequencing, you don't detect it. If you sequence it, you actually see it. But there's if any kind of QC, any suggested guidance for any library prep, you're really completely out of that regime. So that's one thing. And so other things that you see is very large numbers of like primer dimers and things like that, just because there's such low quality and little RNA. So there's a lot of like kind of technical artifacts you have to work on. But we work through a lot of these, and so we developed this kind of metagenomic assay. And so this is what actually the results look like, just to give you perspective. So this is actually, so what we did, we can apply this to a lot of the tests that we did our PCR test on. So this is actually 20 samples, and just to give you a sense of what things look like. So this is, first sample. So you can see that there's about 1.4 million reads. This is the number of reads per, that total. About 1.35 million of them, we were able to classify them. 1.3 million of them are human. 41,000 are bacteria. 2,400 of them are virus. Out of those, 2,180 are SARS-CoV-2. And so then we call that a positive, right? And in fact, was a PCR positive, right? But that just tells you that, you're sequencing 1.4 million reads. 1.3 million of them are human. You're ignoring all of those, right? And you only really have 2,000 reads left. So that tells you that, if it's only because the sequencing has increased by several orders of magnitude is this possible? Because otherwise, like for example, here's a positive with only 104 reads, right? So, but what's interesting is that you see it's concordant with the positives, but then you see a lot of like this person obviously is a COVID negative, but that person has RSV, and this person has RSV also, right? So that's it. So we did a validation, and we showed that it's pretty concordant with the PCR results for SARS-CoV-2, but we see lots of other viruses, and so we see, for example, I guess people ask me like, how much do you see of other things? It's really hard to quantify that. So just in our data, the way that we see is that let's say you have like X positive COVID samples, we see about 0.5x, so have something different, right? So like if, here we have like 87, something like that, of SARS-CoV-2 patients, and something like 45 have another virus. So that's really substantial, right? Of course, this is, at different times you have different levels of virus, but just to give you an idea that there's just, and these are people who think that they're COVID positive, are sequencing, right, are submitting their samples. So there's just a lot of infection that we can detect. But this actually isn't really a good experiment because we don't have a control for, we don't really have a control for the other viruses. So what we're doing, we're actually now doing some experiments where we're going to get data from the health system, and they actually just sent us their first samples, which are they actually run. So they do a lot of testing, and they run these panels that test for 20 different targets, and so we're going to get those samples. So there they have some kind of a positive, like they have a kind of gold standard, classification, which at least what they think are different, what's going on in those. Although they're mostly interested in us applying our technology to the samples that they think are negative, but the person's clearly sick with some respiratory virus, because that's what they're really interested in. So, we're also getting samples from the CDPH also for similar things. But I want to actually, because, CGSI, I want to talk a little bit about the computational genomics component. And one thing, and so this is, remember, I came from human genetics, and so I went into this field, and so I think I have a uniquely different perspective coming into this field. And also, I really want to get things to work, on this project as quickly as possible, and I'm not necessarily tied to any, perspective. So I think I want to kind of share some thoughts on how things work. So first of all, we're using this software called Kraken 2, which I think is really phenomenal. Really great idea. It's been anyway. So this is kind of what it looks, what the results look like. So it actually takes reads and classifies them into the NCBI taxonomic tree, right? And so you can see that, for example, almost everything here gets classified to humans. So like 89% of the reads. But then, when you look at bacteria, right, this is the bacteria. So you can see that I'm not kind of explaining exactly what this means, but you can see that there's like pages and pages of potential strains that it could be. These are read counts. So there's definitely a bunch of strains that it looks like it's, there's more reads, but it's really not convincing which bacteria it's there. So for using this kind of technique, I think for predicting what bacteria samples are there, especially in kind of, poor quality RNA, I think is very difficult. But for viruses, this is actually pretty, this is pretty good. So you can see that this is kind of the tree. So these are the coronaviruses, right? So you could see that this virus is a huge number of, 0.65% of the sample. So is SARS-CoV-2, right? Which is great. We also see a little bit of other coronaviruses, which is also possible. That could either be, because of just sequence sharing between these two and so many, or it could be because there's just contamination between the samples. There's always a small amount. So anyways, that's it. But then you see other viruses also in there, which is as well. So, but one of the things that's really striking, has anyone here used Kraken before for this stuff, right? It's really, really, really fast, okay? Like really fast. And there's other tools that, like for example, looking for contamination, we use like these Kamer counters, right? And they're really, really fast, right? And if you use alignment, it's not so fast, right? If you use like read mappers, it's not so fast. So what's really interesting is that techniques that use minimizers are really, really fast. And these are the relatively new techniques. So I want to just kind of give some perspective on why, because I don't know, how many people here have heard about minimizers? I guess before CGSI, there have been like 3 talks on them. Huh? I just heard about it here, yeah. So I want to give some perspective on why minimizers are so fast. Okay, so what is a minimizer? So 2 reasons. First of all, overlapping, the one idea is that when you have Kamers and you're doing a Kamer analysis and you're looking at every consecutive Kamer, there's so much, when you shift by 1, right, there's so much information that's kind of redundant between the 2, right? For assembly, obviously it's important because you want a longer sequence, but if you're doing other tasks like mapping, you don't get that much by shipping by 1. So like you can, you want to kind of make sense that you don't need to do that, right? And the other thing is that shared Kamers or especially Kamers that have the same minimizer will have similar sequence. So it's kind of an idea that I'll kind of discuss that in a second. But these are, I think, the two kind of key ideas, and I want to give some examples. So, for example, what is a minimizer? So let me explain. So let's say you have a read length of 100. Here's a read. And so the 7 mers of a read sequence are just, if you slide the 7 mer kind of across, right? The minimizer would be the first, from the set alphabetically. Or usually you define something, but let's just say for now, lexicographically, what is the first minimizer? And so this would be the first minimizer because it's alphabetically, kind of before all the other ones. So you can imagine a minimizer as a mapping, a discrete deterministic mapping between a longer sequence and a smaller sequence that uses something like this, right? And so actually here's an example. So let's say you have a 30. Typically you're looking for minimizers of k-mers themselves. So you have like a read of length 100. Let's say you have a k of length 31, which is right. There would be something like 60 different k-mers, right, from that read of length 100. If you take that k-mer and you kind of slide it along, right, you would get, there's 60 different positions, or close to 70 positions that you could start with the k-mer. And so so for each k-mer, you can compute the minimizer, okay? And so the key idea is that minimizers don't change that often. So even though you have 70 different k-mers, right, of length 30, let's say in that sequence, you only have maybe 3 or 4 different minimizers, because most of the time, like for example, if this is your minimizer, it's like somewhere, I don't know where it is, somewhere in this sequence, like it's over here, right? It takes a while for it to be moved out of the sequence for you to get a new minimizer, right? So the only way that the minimizer would change is that either you move out of the minimizer moves out of your window, the current one, or another sequence moves in that's kind of ahead of your minimizer, right? So I think the first kind of killer application for minimizers was if you wanted to count all your k-mers and you're using disk, right? You could then basically separate your kind of files based on minimizer and then write the k-mers to them. And then that way you would have many fewer changes, right? So that was like the original, I think, one of the first kind of applications. But anyways, that is the idea. Any questions on this? Is what the minimizer is? So essentially you have few minimizers change, okay? And so the question is, let's say you want to use a minimizer for not for like k-mer counting, but for like indexing, okay? So let's say what I wanted to do is I want to take my read and I want to build an index for my read. And so this is kind of the, let's say for every k-mer, I compute its position in a sequence, and then I have this data structure where I can look up a k-mer, I can see where it is. So that way, if I have a read, I can look up all its k-mers, I can find where it is, and then I can, kind of map my read, right? So let's say that's like the kind of core, like every mapping approach has something like this, right? So let's say I had this crazy idea, which is that instead of storing all the k-mers, I would only store my minimizers in my table. So I'd only make a table of minimizers. Okay, so for my sequence, I would slide my k-mers across, compute the minimizers, only store where the minimizers are. Right. So now I have a read. I compute the minimizers, and I would look up where that sequence is. So how would that work? Okay. So first of all, what I decided to do was ask ChatGPT to write me a computer program that would do these experiments. So I asked it to write me like a function that would take a sequence length, k-mer length, minimizer length, number of mutations, and it would generate the sequence and then do all these tests for me that I'm going to show you in a second. So I did this, and then anyway, so here's the results of my experiment. So first of all, if I take a million length random sequence, right? There's really two kind of parameters to this. One is the k-mer length, let's say 15, and my minimizer length. So first of all, if I just count the number of k-mers that are in this million length sequence, it'd be basically a million, right? So, but if I look at the number of minimizers, right, it's gonna be a different number. So first of all, it's gonna be at least 20% because there's only 5 minimizers, right, in every one. But it turns out it's about 30% of the positions are minimizers, and this is how many times that they Like most of them only occur one time, you know, but anyways, that's not so important. But the point is that if I was going to build an index out of just minimizers, right, the index would be only 1/3 of the size of the full, you know, set of k-mers, because there's only 30% of the k-mers, right? So that's, and in different lengths, right, of k and minimizers also have very different, you know, like if it's my k is 25 and my m is 15, it's 18%, and it would only take me about 1/5 of the amount of k-mers, et cetera. Okay, so that's one thing. So obviously, like it takes less space to build such an index. The question is, if you use it, how accurate will it be? So actually, let's look at this particular one because this is a good one. So k of 21 and m minimizer of 15, it saves you about a 4x improvement of space. Okay, so then using that program that ChatGPT wrote me, right? What I did was I looked at reads of length 50 with E errors, okay, or mutations, and I computed all the minimizers in there. And then what I did is, so I computed all the minimizers, actually from the read. And then what I did is I added E errors to the read, and I computed the minimizers again. And so if at least one minimizer doesn't change, right, then what would happen is that I'd look up that minimizer and find, you know, the position in the index, and the system would work. If all the minimizers change, right, then what happens is that, you know, it's not like that read's gonna be unmapped, right? So for example, if I look at this, you know, this same configuration, with one error, I only lose 1.3% of my reads, and 2 errors, I lose, you know, 9% of my reads. That's not that bad, right? But still the data structure would be, you know, 4 times smaller. So this is just, you know, you can see that. So anyways, let's just give you some perspective. So that's why, you know, I really think that, you know, these minimizers are real breakthroughs. So I'm very happy that many of you have learned about them. But clearly there's huge amount of research to do on how to make them, how to use them efficiently. Okay. Anyway, so this is a kind of outdated picture of our lab, but been huge number of people that actually worked in the lab and various parts of it. And then here's actually a bunch of the kind of other key people in the project. Thank you. So.